A 22-year-old previously healthy woman is evaluated for skin rash, joint pains, and renal failure. She is found to have decreased C3 and C4 levels and a normal factor B level. Which of the following most likely triggered the complement system activation in this patient? Now, the complement system is going to involve proteins synthesized in the liver. When activated, these proteins can form complexes that can complement acute inflammation. The complement system can be activated through three ways. It can be activated through the classical, the alternative, and the lectin pathway. Now, each of these pathways is going to lead to the formation of C3 convertase, but the way they begin is different. Here's the classical pathway. In the classical pathway, the way the complement system gets activated is through antibody-antigen interaction. Here is the antigen. Let's say that this is a bacteria. And the antibodies we have is IgM or IgG in the classical pathway. Once the antigen-antibody interaction occurs in the Fc region of the antibodies, forms a C1 complex. This C1 complex is important in that it cleaves the complement proteins C2 and C4 to produce C3 convertase. Now that's the classical pathway. In the alternative pathway, we have, it's also called the spontaneous pathway. I like to call it the C3b pathway. So let's write these down. So in the alternative pathway, we normally have autoactivation of C3b. In normal cells, normal cells are going to inactivate C3b quickly. In the presence of pathogens and damaged cells, C3b is going to proliferate. When C3b proliferates, it can then interact with something called factor B and factor D to produce C3 convertase. The lectin pathway is going to involve a protein called pattern recognition receptors, which are going to bind carbohydrates produced by pathogens. Once this binding occurs, we produce proteases. And these proteases are important in that it can cleave the complement protein C2 and C4 to produce C3 convertase. Once the formation of C3 convertase happens, C3 convertase can then produce C3a and C3b. So we can cleave C3 to produce C3a and C3b. C3a is important in that it is going to upregulate mast cell degranulation. C3b is going to be important in two aspects. First, it can upregulate opsonization. So it is going to improve the phagocytosis ability of phagocytes. And it is also going to be involved in the formation of C5 convertase. C5 convertase is going to be very similar to C3 convertase in that it is going to cleave C5 to produce C5a and C5b. C5a is very similar to C3a in that it regulates mast cell degranulation. C5a is also going to upregulate neutrophil chemotaxis. C5b is going to combine with complement protein C6 through C9 to form membrane attack complex. Membrane attack complex is going to form on the cell surface of pathogens and is going to lyse pathogens that way. It can also form on the surface of red blood cells to result in red blood cell lysis, but thankfully we have proteins in our body like DAF and MERL, which can also known as CD59 and CD55 and CD59 proteins, which can stop membrane attack complex to lyse our red blood cells. Now, what we need to know about this question is that autoantibodies can form, and these autoantibodies, what they do is they can bind to host antigens. So the binding of autoantibodies to host antigens is going to trigger the classical pathway, the classical complement pathway. Okay, so it is not going to trigger the alternative pathway. Because it triggers the activation of classical complement pathway, what we see is decreased C3 and C4 levels and a normal factor B level. If the alternative pathway was triggered, what we would observe is a decrease in C3 levels, normal C4 levels, and a decrease in factor B level. Okay, but this is not what we see. So as a result, we know that the classical complement pathway has been triggered, and the only correct answer choice we see is answer choice E, which is IgG antigen complex formation. As we talked about, IgG is one of the antibodies that is going to interact with the bacteria to form the C1 complex to trigger the activation of the classical complement pathway. Answer choice A is antigen binding to IgA. This is going to be false. IgA, IgE, and IgD are not going to trigger the classical complement pathway. The Fc portion of IgA, IgE, and IgD cannot activate the complement system. Therefore, IgA antigen binding does not reduce C3 and C4 levels. Answer choice B is autoactivation of C3 component. This refers to the activation of the alternative pathway, and as we previously mentioned, if the alternative pathway is involved, then what we would observe is normal levels of C4, decreased C3, and decreased factor B levels, which we don't see. As a result, this answer choice is going to be also wrong. And C1 component binding to C1 inhibitor is also going to be wrong. C1 inhibitors remove C1RS from the Fc portion of immunoglobulin and block the activation of C2, C4 by lectin pattern recognition receptors. Therefore, C1 inhibitors prevent activation of the complement cascade and increase, not decrease, complement levels. Answer choice D is C9 lipid membrane complex formation. This refers to the formation of the membrane attack complex, which is at the end, and it is also going to be incorrect.